Good afternoon. I am Dr. Donna Guerin, Executive Vice President of Science and Policy at the American Frozen Food Institute. Welcome to today's webinar, the second in the Sanitation Controls track of AFFI's Food Safety Webinar Series. During today's presentation, we will explore validation of sanitation systems. This afternoon, we are delighted to have Dr. Angela Anandappa with us. Allow me to give a brief introduction of Angela and her background. She is the founding director for the Alliance for Advanced Sanitation and a research assistant professor, Department of Food Science and Technology at the University of Nebraska-Lincoln. She currently leads the scientific and strategic direction of the Alliance for Advanced Sanitation, an organization dedicated to improving food safety with a focus on sanitation. She works with companies to improve their food safety practices and plans, especially focusing on sanitation. The scope of sanitation ranges from facility design and material science to microbiology and chemistry. Prior to joining the Alliance, Dr. Anandappa was attached to Kraft Foods and led supplier and co-manufacturer food safety programs. Before we get going with today's presentation, I would like to turn it over to Sarah Brandmeier, AFFI's Manager of Scientific and Regulatory Affairs, for some housekeeping information.
Thanks, Donna. So we will not be answering questions until the end of this webinar. However, if you have questions throughout the webinar, you may write your questions in the text box on your GoToWebinar control panel, and Donna and I will read them aloud during our question and answer session. Also, this webinar is being recorded, and the recording and presentation slides will be distributed to all of you at the conclusion of the webinar. Again, we are excited to have Angela with us today, and with that, I would like to turn it over to you, Angela.
Thank you so much, Sarah, and thank you, Donna, for the introduction. I am delighted to join you today, and I hope I'll be able to share with you some information that will be helpful in preparing sanitation programs and especially looking at validation. So I'm very excited to join you at this particular time, I'd say, in our history here in food safety, because sanitation is really taking center stage in terms of how we look at food safety in the facility. And I think with the new materials that AFFI has produced, we have so many things to look forward to and so many resources that the frozen foods manufacturers can really look to do a lot in terms of improving their food safety programs. So I'm hoping that this event and this webinar will give you a couple of resources, and also knowing that the topic of validation of sanitation is potentially new to some, and maybe one that you're still trying to grapple with how to do. So let's get started. Just as learning objectives, I know today is a day when we're trying to see, what do we want to do in terms of sanitation validation? What does that mean to us? So I'll provide an overview of validation. What does that mean for sanitation specifically? And then we'll talk about understanding the importance of validation in the context of sanitation. We're already familiar with it in some other areas of the facility. And then we'll talk about some strategies to review and look at validation studies. I'm particularly interested in this area because it gives us a view into how to look at validation programs, especially those which are maybe not ours. And then in situations where we do need to develop our own, how do we go about doing that? We look at some of the pitfalls and then look at effective strategies to target and manage Listeria monocytogenes. So with that in mind, I'd like to just go over some of the definitions that are in the CFR, just to remind ourselves of where we really are. So validation in terms of a food safety plan, we're used to seeing validation in terms of process validation, and it takes the form of thermal validation. It takes the form of
dialing in again.
I'm sorry, did we?
There we go.
Sarah,
you're back. Perfect. Thank you. Yes, thank you.
Okay. So I'll repeat that just to be sure that we didn't lose anything. But I'm trying to review, the terms that are relevant to food safety, validation, verification, monitoring, and microorganisms. So these are terms which we'll use today and that we are familiar with. We've all heard about these. We know how they are used. We know how they're used in our HACCP plans. We know how these are used in our preventive controls plans. And then today we'll look at it in the context of sanitation. So validation means obtaining and evaluating scientific and technical evidence. So we're trying to make sure that the system we have designed is going to work every time correctly, and that's validation. Verification is the application of methods and procedures, testing whatever our systems are that are appropriate for that system to monitor and make sure that that system is working frequently and correctly in accordance with what has been validated. Monitoring has various things attached to it. And I guess, in terms of whichever process you're looking at, monitoring can look quite different. The other is microorganisms. Today, we're going to look specifically at Listeria monocytogenes. Of course, there are several other organisms of concern, which may be specific to each process, but we'll look specifically in the context of Listeria monocytogenes. So validation, verification, monitoring, and then microorganisms, and making sure that we are looking at the right microorganisms for the process are key factors in validation. So why do we care about this? This is not published data. We're still working on some of this, but I want to share with you that approximately 50% of recalls can be traced back from a root cause perspective to sanitation. So there's a whole lot of incentive for us as industry to really look at sanitation and be able to improve those sanitation systems so that we can prevent recalls. And there's so many aspects of recalls. Primarily we have the financial aspect, the food waste aspect of things, and of course, brand protection. Between those three, I think there are huge incentives for the food industry to really look at minimizing and eliminating the potential for recall. The other is that sanitation is a huge cost on our energy systems, the cost within the plant itself. And we know that it's something that, most companies are looking forward to seeing how they can minimize those kind of costs. It may be utilization of energy, it may be water, it may be various other aspects, including, the human resource aspects of conducting sanitation activities. So sanitation is a definite target in terms of how we manage food production, how we manage our processing environments, and how we are efficient in producing food. The other is, employee turnover is huge in terms of sanitation. We know that that's one of the big challenges in the food industry right now. And so we want to make sure that when we are looking at sanitation, we're looking for ways in which we can improve sanitation holistically for the business to improve. We also know that outsourcing sanitation is a bit of a challenge. Although many companies are moving in that direction and third-party sanitation companies are really, really growing their skill sets, it still hasn't been able to, really solve all of our problems. So it's another area in which we're looking to improve how we do business. So sanitation data is where I see all of this, playing a role, that as a business, you want to look at this as sanitation data and how we improve that. So validation means we can look at all of these very big topic level areas that we can improve. And then validation of any one of those is a debatable topic, but it's something that we can. So let's look at validation in the context of regulatory environments. Validation is required, as you all would know, for process control. So preventive controls, your hazards are identified, and your hazards in terms of chemical, physical, biological hazards, radiological hazards, we need to control them for using process controls. We're all familiar with that. When is validation not required? And we see that if we look at the CFR, if we look at, most documentation, sanitation controls are required. However, validation of those sanitation controls isn't something that we are required to do by law. Neither are we required to do validation of supply chain controls, although we know foreign supply verification and a supply chain control, supply chain applied control is important that it is validated if we are the consumer of that product in our facility. We also do not need to have our recall plan or allergen preventive controls validated. So if you look at that, we see allergens, we see potential biological hazards, all that do not necessarily need validation, but they do need control. And FSMA really tells us we need to be having those controls and having confidence in those controls. So when is sanitation validation something that we need to look at? And I think in the context of frozen foods, and I think for AFFI's membership, this is a really important topic that we need to understand why and how it's to be applied in our facilities. So sanitation, in terms of having it in our preventive controls plan, often becomes a way in which we say, yes, I'm controlling certain biological hazards using my sanitation program. I'm also going to make sure that the sanitation program is preventing various chemical hazards, various allergens from being able to get into my product and contaminate it. So I've identified 4 specific areas here. I think there are others that could also fall in here, but I think, as a best approach for looking at sanitation in this context, validation would be almost always the best practice that we can adopt when sanitation is the preventive control measure that we're using for managing Listeria, managing any kind of contamination that occurs after our heat step, and for ready-to-eat products that do not encounter any other control, including some specific or packaging-related controls. Sanitation is also perhaps the only break between product types, allergen-containing products, or various products that are manufactured by co-mans between brands and between product lines. So these are areas in which a validated sanitation program would be really critical to be able to get the results you're looking for. And if you look at the entire Listeria control program that AFFI just recently rolled out, you'll see that there are 6 specific areas that are very well addressed in that program. Of those areas, sanitation controls and practices are the one we're talking about here. And this has to work very well with the other controls and the other plans that you already have resources. So I'd say, today when we talk about sanitation validation, let's look at that in the context of this entire system in which we have GMPs and personal hygiene as a key part that we've already addressed. Controlling the use of water. Perhaps we need water, or we do not need as much water, or we need no water in our facility. So all of those are sometimes differently applied depending on our process. And of course, we're looking at the hygienic design of facilities and equipment, that is in really good, strong condition. And then of course, freezer management, freezing in general, and environmental monitoring, that we would have a good, strong program. So these need to work together and have our sanitation controls working very well with them. So what are the benefits of validating a sanitation program? And I'd say there are so many benefits that you can look for, but the top one that is of benefit to a company that may not be really looked at from only food safety perspective is extending our runtime. If we'd like to run a product for a longer period of time, let's say you're running for 6 hours and you're cleaning right now, and you'd like to say, I'd like to run this for 8 hours. That's the kind of benefit that we can get from validating a sanitation program and having the data to back that up. We can also use a validated sanitation program to compartmentalize our critical control points and be able to troubleshoot issues that may occur separately from one another. Now, that does not mean that your entire sanitation program should not be looked at, but it does help you look at validating parts of your sanitation program, which I think many companies like to look at as unique entities within that program. And it's useful to do it that way. You can also have confidence in controlling the hazard that you have identified. So Listeria monocytogenes, which is our target organism we're looking at today, is one of them. However, you might want to identify certain other organisms that are of concern. Certainly, there are some viruses that we still need to do a lot of work with, but we do have to look at E. coli species. We do have to look at salmonella sometimes, but Listeria is a huge concern, and specifically for frozen products, we would like to make sure we have none that enter the product. And then documenting and demonstrating control to inspectors. That's a huge benefit that we can get from having a validation program. We'll get to that a little bit further and look at what that consists of. And then using validation to give us the kind of data we need to make broader food safety decisions. And I'd like to call that the part of the database decision-making process that we all need to have. That is really robust. And of course, it's difficult to obtain that if you don't have validation data to go with it. So as a quick overview, what is a validation study? And I'd like to go through, what would you want to put into a validation study? So designing the study, who is going to be designing that study? Is it an internal or external expert to design the study? And then identifying the study objectives. Why do we want to do a validation study? And again, if we think about this, not from a process perspective, but from a prevention of contamination within the environment perspective, then our objectives look a little bit different than saying, I want to thermally remove Listeria in my product. Now I'm assuming that my product itself, whether it came into my plant containing Listeria, or whether it came in not containing anything, we've already got that maybe addressed by our suppliers. But still, within our plant, when we are having certain processes being undertaken, or our product needs to be, it's a mixing process, whatever that might be, to ensure that contamination of the product is prevented because sanitation has been well addressed. So the objective of our validation study then is about making sure that our cleaning process is really running the way it should. So the target organisms or allergens, those which are typically our targets for a cleaning process, are really what we're looking for. And then the scope is a huge debate, I'd say, here, because looking at cleaning an entire facility, cleaning a piece of equipment, cleaning 2 or 3 pieces of equipment, cleaning the floors and ceilings, all of those are pretty discrete when you think about it, but they rely on one important thing, and that is your sanitation standard operating procedures. If your procedures are not very well documented and very well designed to address the needs of that particular target or piece of equipment, the entire validation really has no meaning. So we need to have validation built on good SSOPs that we can follow. And so this is procedural. If you really think about it, the whole validation is going to depend heavily on not just the functioning of the chemical or the process by which we conduct it, but also the people that conduct it. So our SSOPs are really important. So sanitation program elements, which are the who, the what, the when, where, and how, are all going to be elements you're looking at in terms of validating the sanitation program. You're going to look at chemicals, and chemicals used for cleaning and used for sanitizing are different. So we need to really view cleaning as distinct from sanitizing. This is an area that I have seen many companies, many professionals struggle with. The mere use of the term sanitizer or sanitizing sometimes means cleaning, applying sanitizer, and it sometimes means both. So for the purpose of our sanitation program, having it clearly understood by everyone who's performing the activities that sanitation really means a robust method by which you're going to clean and fully clean the targets you're looking for, is distinct from the application of sanitizers. So a 7-step sanitation process, which we will get into in a bit, will require that you are going to take down your equipment, you're going to do these inspections, you're going to do the cleaning, you're going to have all of these steps by which you can follow the 7 steps of sanitation. But in addition to that, you also are making sure that that everyone who is conducting those procedures is going to conduct it in the same way. So validating may simply mean for some facilities that every employee who's practicing those SSOPs for equipment, you will be able to measure and be able to see the variability between those different employees. And of course, you're trying to make sure that if there is an error at some point in the process, may it be the application of chemicals or the time that for which you would be applying those chemicals or scrubbing, whatever that might be, agitation is in a CIP system, for instance, right? Whatever that might be, that you are going to be able to determine which point you would have a failure at. If you might have a failure at 3 minutes of contact time under these conditions, you want to know when that's going to be so that your system is validated to work despite that. Let me go through what that might mean for your suitability of the operation. So here we're looking at the different components that it takes for the operation to work. For infrastructure, you might just look at the design and the maintenance and the layout and all of the facilities where it might be. So let's say we're looking at an agitator, and you want to make sure that cleaning that agitator in a certain way is going to be adequate for it to be it to not carry over allergens or not carry over Listeria to the next process. So that might be your target. You might also want to look at validating that the chemicals work in a certain way, and that's going to be adequate for that particular operation. We know that that appropriate cleaning and appropriate sanitization really requires both the application of chemicals, the correct scrubbing and protocol in which they're applied, and for the right amount of time. So all of these working together is what makes our sanitation program really work. So what we are measuring is effectiveness. Is the program we have designed to clean our equipment and sanitize that equipment achieved? Is it going to be effective in achieving the elimination of Listeria, or prevent Listeria from having any contact with our product? So in terms of how we do so, we're looking at some level that we determine is appropriate for that. Is it visually clean? Is it an allergen clean we're looking for? Is it a pathogen clean, or is it a sterilized clean? So those are 4 different areas or 4 different ways in which we can look at our target. So if it is cleaning equipment that we want to validate is cleaned, perhaps we look at it from, do we want it to be sterilized? do we have a system by which we're going to sterilize it? The picture I have here is of a cabinet washer. We want to make sure that cabinet washers, we're actually going to do a webinar in a week on that through the Alliance. So if you'd like to join that, you can just let me know as well. But how do we make sure that we can validate the cleanliness of clean out of place equipment? And so the cleaning protocol we're looking for the equipment itself would be something that's determined by the system we're using to clean that equipment. So together with validation, what goes hand in hand with that is verification. And that's ensuring that the system is working properly. So validation of a system that would eliminate potential Listeria in the facility means that we also need to have the appropriate verification method to ensure that we're getting the right results. And then we would want to build in a monitoring system for that, and then documentation. Very similar to HACCP, very similar to preventive controls for a process control. But similarly, we do that for a validated cleaning step as well. So in practice, we're looking at the various methods, so many of them, to ensure that we can test, be it ATP, or is it a swabbing sample that we would send out to a lab, or is it an internal test that whatever our method is that we would want to use to verify, we make sure that we're documenting information, we're able to review those records and reevaluate that. So let's look at validation and what the validation needs to have to be successful. And I'm taking this into the perspective of going into a suppliers or co-manufacturers, and we're trying to work with them. We want to make sure that they're going to provide us with the kind of validation that helps us have confidence in their process. Similarly, this may be your plant in which you're trying to develop a validation study that is going to provide confidence to your customer or to whoever your stakeholders who would want to be looking at that, including auditors and inspectors. So the objectives of the study need to be really well defined. Is it the entire system we're looking at? Is it a piece of equipment, or is it multiple pieces of equipment? What is the scope of that? Is the data supporting the outlined objectives? So the objectives of the validation study, if it's determined that I want to make sure that there will be no contamination by Listeria at the end of this cleaning process in this piece of equipment, well, then the data needs to support that and not something else. And of course, how do we know that the Listeria is under control? So let's look at some examples in which this might be useful, right? And if you were to develop your validation study and your objective was how long can the process run between cleanings? That may be the question you're asking, or the question that validation study is trying to make sure that it addresses. And like I said earlier, do I want to extend it from 6 hours to 8 hours? So let's say that's our example. So we want to make sure that the study is going to give us parameters for 6 hours, parameters for 8 hours, and potentially longer, in which we can be confident that running our process for 8 hours would provide us no more potential contamination by Listeria by using the same cleaning protocol. Now, of course, to do so, we need to know when do we break the system. In other words, what are the limits? We're familiar with the term challenge study. A challenge study is one in which you're really trying to see how far can I stress the system. In many ways, validating the piece of equipment and its cleanliness can be looked at in that way, but these are two different activities. Validating for cleaning is not necessarily the same as validating a process, but validating for cleaning also then means I want to make sure where my breaking points are. So let's say our chemical supplier somehow dilutes the chemicals. We hope no one's doing that, but if or an operator or somebody is either using excessive amounts of chemicals or less chemicals, does that have an impact on the presence of Listeria? Those are the kind of things we're looking for in validating. And we want to make sure that, you know, the parameters that we're looking for, be it the number of organisms that are present at the end of the study, or the number of ATP or ATP, let's say we're looking for ATP only and not even Listeria, right? That's going to be something we're looking at in terms of monitoring. How does that relate to the presence of Listeria or the presence of E. coli? Do they have some correlation? Does our validation give us some confidence in which we can say, okay, at the level of you know, bioluminescence for ATP, let's say we're looking at 10 units. Does that correspond to the presence of Listeria? It's a little difficult to easily determine without having a good study that supports that. And of course, how do we have how do we use surrogates when we need to do so? So assessing the effectiveness of the sanitation program to address Listeria monocytogenes specifically, and if Listeria is our target organism, we need to include several pieces of our sanitation program in the validation. I would include my my periodic equipment cleaning, my pick and pack program, to make sure that not only am I including my routine cleaning of the equipment and the facility, but also that periodic equipment, periodic infrastructure cleaning, making sure that there are no niches that are just going to be propagating some Listeria. There are multiple ways in which we can do so. There's already strategies, and I would really refer you to say, you know, go to the resources that AFFI has already created. There's so many wonderful resources there to look at how you'd want to do that. And then, of course, the program elements within the sanitation program itself, which is, again, cleaning, sanitizing, and then looking at these in distinct ways. Looking at cleaning, definitely distinct from sanitization, and also making sure that we are having a clean facility before we say, yep, we can apply sanitizer. And the routine sanitation protocols, SSOPs, if you have it. So some pitfalls, and we know that recent recalls tell us that there are several issues that may be occurring in the facilities that we might put with. How well we're looking at data. We can look at Listeria draft guidance to give us some ideas of what FDA is expecting from us. And then, of course, I want to point to a couple of things that you can easily go in and look at your facilities immediately. That recalls prove there's failures in employee practices. I think that's common knowledge. And of course, scheduling failures in sanitation. This is a tougher one to look at, but scheduling in sanitation really has a close relationship with some of the recalls that we can see right now, or that we've seen in recent years. Chemical mismanagement, run time. You know, how long can we really run without having a clean break? A clean break means we will not have, you know, thousands and thousands of pounds of product implicated, right? Not certainly millions. So having those clean breaks and being able to identify when the clean breaks are is really critical. SOPs for each that people follow. And I can't emphasize enough how important it is that SSOPs are followed by every employee and every system that we have designed consistently. And these are failures that really have much more to do with how well we train our employees, how well we manage those employees, and then how much ownership they have to make sure that every activity that they conduct is going to be conducted in the best possible way. And then let's look at some of the pitfalls that you might encounter. The number one thing, say, easy to fall into, not really, is just including proper inspections before sanitizer is applied. Not using, I'm sorry, that's a typo, not using a well-designed and executed environmental monitoring program. Are required by SIGMA, but in addition to that, they are a huge source of knowledge for us to see and then be able to advise our sanitation program if you use it as well as we can. Misapplying and failure to conduct cleaning verification. And verification is often confused with monitoring or your EMP program. Also with validation, it's not uncommon for people to not understand what cleaning verification is. I've personally seen it so many times where cleaning verification is not something that's done in advance. Let's talk a little bit about that, because cleaning verification is sometimes potentially possible just visually. You can look through and see, you know, is it allergens I'm looking for? Am I making sure that there's, you know, no visible residue? Is that adequate? Is that enough for that particular process? Sometimes it is, sometimes it's not. Maybe it's a dry process. There are certain dry processes in which, you know, that visual inspection is adequate. But then there are others in which having clean equipment swabs done, and we would want to do so after cleaning. We do not want to collect clean equipment swabs after you've applied sanitizers, which is a mistake that many make. And that's because swabs themselves may be containing some of that sanitizer, and you won't get a positive if there's a positive. There's also plants that sometimes swab, but they're swabbing for negatives. They're looking for the negatives. You should be looking for the positives. When you have a positive, you know what went wrong, and you can fix what went wrong if you know there's a positive. So these are easy pitfalls that people fall into, and they are ones in which you should be constantly, you know, looking to try and fix. Improper sampling. Are you using the right zone, amount of sampling area? Are you swabbing appropriately? And then have you appropriately handled that swab before it's sent out to the facility for analysis? Is it an internal lab that's going to do it, an external lab? Have you passed on the right information so that the lab can process your sample appropriately? And of course, a validated system will give you certain benefits that you can take in a way that you can also, you know, turn it back into optimization of your system, optimization of your water usage. And an optimized system means it's going to be effective. You are effectively eliminating your hazards, and you're also going to be efficient. So this means, you know, you have the benefits that you can say, yes, I have, you know, cost benefits. I'm able to run things more efficiently. I'll have less downtime, and together it makes having a validated system a financially viable one as well, and one that's beneficial. So measurement systems are important. What is it we're using to make sure we're measuring in order to get those gains? We need to identify the right targets, the right cleaning protocols to eliminate Listeria. And of course, some of these you can obtain through your chemical suppliers. You can collaborate with others in the organization to make sure that you are doing the right processes for that particular product. And then, of course, using the right indicator organisms. Coliforms are commonly used as indicated organisms, but for Listeria, Pseudomonas is a really good one to use as well, because they tend to be in the same kind of biofilms. And then when to measure, how do we measure it? and use the right protocols for doing so, use the right kind of swabs, using the right kind of gather the right information, and in the right locations that you'd identify in your swabbing plans. To understand the detection limits, especially if we are looking at the detection of microorganisms, we're looking to see, am I looking at one CFU, and then what area am I looking at? Did I capture that one CFU and one microorganism, one cell, for 10 inches by 10 inches of a swab? What does that mean? How do I interpret that? Things like that. And then, of course, use the right method. We know that FDA and USDA as well, they're swabbing and they're doing whole genome sequencing. How do I use that? And is there a better way in which I can get that kind of information? We have started using metagenomic systems by which we can look at much more of what's going in our environment. And we're trying to see how we can better project what might be in that environment using metagenomics, which isn't the same as looking at whole genome sequencing. And I'm happy to share with you any other information on that if you have specific questions as well. And then, of course, the infrastructure by which, in which our plant is operating. So we want to make sure that at some point we are also collecting some data and making sure that, our validated system, our validated cleaning system is functioning in a larger system that's functioning well. So it's a whole system that we're looking at, and that whole system, it comprises of so many areas that we can actually validate and we should validate. How do we pick which one we really need to be looking at and we really need to validate to control Listeria? This is a challenge, and this is also what I think you need to look at in terms of your specific operation. This is a quick example of a study that we did, and we just used different cleaning protocols to see what's the most efficient. Now, you can do something like that in your own facility with your own cleaning protocol for that specific environment to see what's the best way in which you can take out a specific organism. We use Listeria inocula. You can do something like that as well. And of course, you're looking at how efficient all of this is going to be. So the efficiency and the effectiveness, take that together and be able to make a decision as to what is the most useful data that you can use. You monitor this and you're going to trend this data and see, was it done right? Was it done right the first time correctly? If it was not, how do I fix that? So you take all of this information, and then Listeria control is obtained once you can take your cleaning verification information, your trend analysis, which I just mentioned, the information from your validated sanitation program, your environmental monitoring program, and combine this with the very well-designed SSOPs that you're going to use to conduct your cleaning processes. So I want to leave you with the idea that Listeria control means you're going to take all of this data and be able to trend that and look at that and be able to make some decisions. So with that, I will stop. And if you have any questions, I'd be happy to answer them.
Thank you, Angela, for an excellent presentation. I do appreciate that. I'd like to open up this session for questions. Again, you may write your questions in the text box on your GoToWebinar control panel, and Sarah and I will read those aloud. While we're waiting for that, maybe I'll ask you a question, Angela. In regards to resistance that can develop potentially with pathogens in the environment, how do you best advise on trying to reduce that risk of antimicrobial resistance in, pertaining to sanitation systems?
Yeah, it's a really good question, Dana. And I think it's a question that people are battling in terms of how do I manage my sanitation program and how do I make changes to that? One of the ways we can look at in terms of what's going on with that is really looking at, changing our sanitation protocol from time to time. But again, that's an interesting question because, we want to do that. Innately, we know that there's going to be potential resistance that's going on, or more like persistence of the organism over a period of time and then changes. But we can also track that, not only using our organisms of concern, so let's say Listeria monocytogenes, but what I like to call Listeria's posse. But because Listeria has its own organisms that it likes to hang out with. And there are helper organisms that like to behave a certain way with Listeria and behave a certain other way with others. And we also know that over a period of time, these biofilms or organisms that like to hang out, together change. And then these are things that I think each plant having to monitor on their own is in a sense unreasonable if you have to culture all of them. So this is one of the reasons we're looking at metagenomics as a potential use for us to be able to see what's happening longer term in terms of changes. So that if you are able to see a change metagenomically in that facility, and I know we're getting into almost a different topic here, but if you're able to see a change in the entire population, you might want to then change the sanitation protocol that you have. Maybe you use a different sanitizer, but perhaps you're looking at really targeting your cleaning protocols first rather than your sanitizer itself. And I think that's one of the key ways in which we can prevent or minimize this sort of risk that you'd have in developing resistance is by, having adequate time for your cleaning protocol to have done its job before you use a sanitizer. And then once you use a sanitizer, ensuring that you do not run product immediately. You need to have a specific amount of time after that sanitizer has been applied for it to do its job and be effective and take out any residual organisms, and then be able to run product that doesn't get contaminated with sanitizer. So I think timing is really key to doing that, and the more time you can build into those buckets in terms of cleaning, sanitizer, and then running product, the less potential issues you might have, and also using more data, such as that metagenomic data, to be able to make decisions as to when you need to change your sanitizing protocol.
Perfect. Thank you. That was great. And like you said, we could do a whole nother webinar on metagenomics for sure. So we have one question that is more about, they want to see the efficiency slide again. But we can actually, for those of you on the phone, we're actually be sending the presentation to everyone after this call, and so you'll get all the slides in addition to that one, as well as the recording. So can you read the next question? I have a hard of seeing issue right here with the small print.
Sure. So Angela, our next question is, the ATP test, does it help to validate the effectiveness of sanitation?
It's a good question. My quick answer to that is no. And that's because ATP is an interesting system by which we can get a lot of quick information. ATP is wonderful because it gives us very fast information with plottable data, right? But also it gives us really pretty information. If we have tons of it, we can get. It's also rather cheap. But ATP does not really tell us whether or not we have living organisms every time. Because ATP alone tells us we may have free ATP around. We might get that. You actually can get higher ATP readings when there's no organism, but maybe product residue there as well sometimes. So it's not necessarily the best indicator, I'd say, of a validated clean. But it does give you an idea of how well you clean rather than how much contamination you had. I would prefer to use ATP to tell me, did I do a good job of cleaning? And if I expected to get this level of clean after this cleaning process was done, can I get a quick reading of did I do a good job before I say, okay, now I can go and apply my sanitizer.
Great, thank you. Our next question then is, what is your top 5 organisms to monitor when validating a sanitation program? And does one organism suffice?
I think for this audience, especially keeping in mind that, you know, we're really interested for frozen food in not having Listeria there, I'd say Listeria would be top on my list. I would want to look at certain species of E. coli, especially the ones that are enterohemorrhagic. I would want to include some of them. I'd also be interested in Pseudomonas, because Pseudomonas is one that, as an indicator, is telling us quite a bit about the biofilms that are present. So I would include Pseudomonas. It is not included by many people, and I think it's very much a debatable one, but I would include those. For frozen, I mean, certainly there are also viruses that I'd be interested in. It's very hard to do, but I think it's one that's on the horizon. Let's say 3 years from now, I'd like to have some way in which I can also look at norovirus. So I'd say if you're asking for 5 organisms, that you may not need 5, but you certainly could use a few indicators there. You certainly could use your pathogens of concern, and you should be good.
Great, thank you. Our next question is it better to do swabs before or after sanitizing?
Before. You need to do your swabs after cleaning. Ideally, I'd like to look at your sanitation program as heavily relying on your cleaning protocol rather than your sanitizer. Because your cleaning protocol really is the one that's going to take care of any biofilms that are there or any kind of niches that you really want to address. And how well your sanitation program is going to function is going to rely heavily on how well you actually clean. So once you've cleaned, if you take your swabs after that and you're making sure that it's actually clean, that's the right time in which you're going to get the best data with no interference from a biocide of any sort.
Great, thank you. Our next question, would you revalidate your sanitation every time you change your process? And the example that was given with the question is different products.
I think there's different schools of thought on this, but in general, I think we're trying to look at this as not necessarily dependent on the product, but dependent on the cleaning procedure used. Cleaning procedures will rely in some ways on the type of product. So if you're having a very heavy, fat-heavy product that you're running, you're going to use different cleaning protocol than, let's say, a bunch of pressures, right? However, if you are using the same cleaning protocol and you know that it's going to be adequate for, 10 different processes that you're running, 10 different products that you're running, you should not need to validate it between each one of those. You should be able to have it for a change in your cleaning protocol only.
Great, thank you. Our next question is there recommended validation metrics to present to a regulatory inspector that will assure clean break?
I think that's, I'm not sure I'm understanding if you're asking for like a matrix you can refer to, because I don't think we really have something like that developed, although I think that would be wonderful. But for your plants, you might want to have something like developed to show that, yes, you have a clean break from time point A to B, and then your inspector will be able to say, yeah, okay, that is indeed a clean break. So what does that clean break comprise of? That's the tough question that you need to really determine. And I think I had several slides there that you could probably refer to, but if I didn't answer your question, please feel free to email me or just call. I'm happy to try and answer this.
Great, thank you, Angela. So the last question that we have here is the asker of this question had an experience with an auditor who almost gave them a nonconformance for not taking into account spores in their EMP and sanitation validation. So what is your opinion on this?
So spores, I think, are tricky. They're not, you can look at that in certain processes. If I was in a dairy plant, I would be wanting to look at some amount of spores that people are looking for. But spores are spores, and they're really tricky to be able to capture. I think it would be very risk-based, very much dependent on the type of process you're running. What is the output? And then, there are certain processes in which it would be very helpful to have spores captured somewhere.
Great. Is that all the questions?
Yes.
Great. Fairly robust Q&A, so really appreciate that. A lot of interest in this topic, Angela. So again, excellent presentation and participation today. I think we do appreciate you highlighting some of the resources that AFFI's created. And those were created in partnership with our members. So they're very much from people's programs and reflect kind of what's going on in the industry right now in regards to best practices. So appreciate that. So I'm going to turn it over to Sarah, who will close us out for today.
Thanks, Donna, and thank you again, Angela. And thank you to everyone for joining us today for our webinar. And as was mentioned previously, the webinar slides and the recording will be distributed to all of you at the end of this webinar. So thank you again for joining, and have a great day.